this semester. There's very few points in time that I'm not in a lab or in class. So if you didn't make my 12 to 2 today, then you might be a block between now and Wednesday. But certainly you can email me, and as best as I can, I'll try to reply in a timely manner. Your homework was due, so you guys don't have that in your hands anymore, but what I'm going to do is right after I walk out of here, I'm going to get on my computer. I type like this. Get on my computer and upload the solutions for this homework. So you guys will have that directly whenever I get home from now. Okay. So you'll have the answer to the solutions that you can seek out from there. Okay. So the stuff we have to get through tonight that's in terms of new material that's going to be on the exam is we're just finishing up this tetrad analysis. So we started talking about neurospora, we started talking about yeast and how they can stack their meiotic products and put it all into these little spore stacks. So we're finishing up that discussion tonight, and then so after that, that's nothing new for Wednesday. Okay. And what I did is I'm kind of making a compromise between those folks who might want just have a few questions on the homework or other sorts of office hour type of formatting, and then those folks who actually wanted me to just kind of like hold your hand a little bit better and do some more practice problems. So I made up a couple practice problems. One is a pedigree type of problem, something we haven't actually looked at before. So a little bit of new working around with use a set point example. And then the second one is a chi-square. So it's actually taking incomplete dominance and recessive epistasis and comparing, trying to determine whether your data fits that analysis. So we sort of did an example similar to that earlier, and we talked about how you would do breeding studies to if all you had was breeding information. So then you can apply a chi-square analysis and really decide between those two possibilities. So that's all I have prepared. And then if we have any time like left over in the lecture, that's going to be more on you guys if you just have quick questions, office hour type of questions. I'm not going to work pre-made problems for you type of thing. Okay. All right. So before I get into tetrad, any questions from you guys? So I'll finish up tetrad, and then we'll try those practice problems, and I'll say a couple of things about the exam before we get in with that, but we'll finish up tetrad first. And I know I said earlier, which is true, that there are sort of this idea of ordered spores. Remember we thought about ordered versus unordered. So whether those spores are kind of just present in a sac or whether they're kind of in this lined up, stacked, ordered fashion, right? And you can use both of those species of yeast, single-celled organisms. You can use both of those to do mapping analysis and genetics, okay? Your book, I'm pretty sure it definitely walks through an unordered example. However, so I was going through and I thought about the timing of all this and decided to give you guys sort of a break. I know I said on Wednesday we were going to go through both of these. Okay? We're actually just going to cover this one for the exam. So for your study purposes, I'm planning to lay off you guys a little bit, not give you so much new material on Monday. So all you need to know for Wednesday's exam is how to walk through an ordered spore sac analysis. Okay, that's what we're going to do right now. Okay. So an example of data that you might see. So these are all going to be octads. Okay. So it's a form of tetrad analysis, but that tetrad, that product of meiosis, remember those 4 spores in the sac, undergo mitosis, they get a copy. So you have an exact copy of each other. So now we have 8. Okay. So you get, usually in these types of problems in ordered spore mapping problems, you get a set of data and it's showing the alleles. Okay, so remember we drew up the chromosomes and thought about if there's a crossover that happens between the centromere and this gene, then we end up with one certain type of order of spores, right? So we had this idea of either having big A, little a, or you could have this alteration going on here, right? Depending on whether that crossover occurred between the centromere and the gene or on the other side of the gene. Because this whole idea of mapping tetrads for ordered spores, what it gives you is distance from the gene in the centromere. So we're really asking, if we just keep it as gene A, make it simple, what's the distance of gene A to the centromere? That's what the kind of data that you're going to get from this. Okay. So here's what you would be presented with. So you're going to be given essentially the spore genotype, and then how many numbers of that, those types of patterns that you're collecting in your data after sexual mating between yeast. Okay, so they undergo meiosis, they create the spores, those undergo mitosis, so we end up with an octad, and you're getting gamete information. Okay, and you're getting it in terms of numbers of individuals that show this. So underneath this, we'll have, say, 126 of this style, 132 of this. So we're still keeping with gene A.
Yeah, spectral column is supposed to be another 8, right?
There's only 7.
Thank you. My eyes start going cross-eyed looking at all this. All these babies. Yeah, you would always expect to see 8 recovered. So we have a total here of 300. So what do we see? Pausing to let you guys record all the A's down. What do we see in terms of any kind of pattern here? Just by looking at it, thinking about what we talked about with the chromosome patterns, the result of whether you have a crossover between the centromere and the gene or not involving centromere and the gene. Anything standing out? Right, so automatically if you just focus on the numbers and not even the gamete genotypes, we see kind of a similar thing than what you guys are used to looking at with a 3-point cross on a multicellular eukaryotic typical diploid organism, where your largest numbers probably are going to indicate some kind of no crossover event. But the thing that's tricky with these spores is it's more that it's indicating a no crossover event between the gene and the centromere. Because it's much less likely, remember we have our centromere, okay, and if we have gene A here, a crossover anywhere along the length of this chromosome is at any location is going to be much more likely to occur than one to occur right exactly where we're in a narrowed down spot in a smaller distance. We're asking for it to be just between A and the centromere. So now you're looking for a least likely probability because you're focused in on a certain distance. You're telling it could be anywhere on the chromosome, or what we want to see is recover these ones that have had this recombination event occur between the gene and the centromere. So that's always going to be less likely to occur. So these two larger numbers still indicate there was a crossover event, but what can we say about the location of the crossover event? In terms of, yeah, so on the other side of the gene, and the centromere itself. So it's the crossover event's either happening up here, the top arm of the chromosome, or it's on the other side. So it's not in this window that we're kind of focused on right here. Because remember also, if you look at the pattern, so not just the numbers, but if you look at the pattern, all the dominant alleles are stacked together, all the recessive alleles are stacked together on those far two left categories, right? But these two have a very different pattern of alleles than the other four categories that we have up here. You either have all your dominant alleles stacked together, and that matches this, right? What we drew over here. Right, so if you go back in your notes, what produced this lineup? Big A, little a, little a. Just on this one from your notes from Wednesday, when you guys had
Which chromosome?
It was on the other end. So this isn't, yeah, you almost don't need to think about homozygosity and heterozygosity for these types of problems. But Melanie's right, yeah, so it was when the crossover occurred down here. Yeah, or technically it could be at the top too. Anywhere that's not in that window that we're looking at, right? Mm-hmm. Okay, we drew out our little chromosomes and we showed that this was producing that event. Okay. So this means you have no crossover, right? Between the gene and the centromere. There's still a crossover, it's just not in the area that you're interested in. And conversely, this pattern, when we get this alteration, if you look back in your notes, we drew the chromosomes out. We thought about what that would result in after meiosis 1, meiosis 2. This means you have a crossover between, and it's falling in that window space. So this one's falling right somewhere in the vicinity between the centromere and gene A. So this one is occurring anywhere else in the chromosome except for that spot. And then when you alternate alleles in any fashion, okay, then that means that you've had a crossover that's actually occurring between the gene and the centromere itself. And then you just undergo mitosis, and everything stays in an ordered ascus. In an ordered ascus, you have spores, and they stay exactly where they are. So even when they make a copy with mitosis, after the meiosis events have happened, all we get is a doubling of each set. So that's why you can still see 4 dominant A's together, 4 recessive A's together. And same with these other 4 classes of gametes, of spores that you've recovered. They're just duplicating, right? So this is still big A, little a pattern, but you just duplicated each allele. Okay? Each cell, or spore, I guess I should say accurately, makes a copy of itself. Okay. Does everyone see that patterning at least?
Hey guys.
Yeah.
Maybe I'm missing. So maybe I'm missing the background. So you have the haploid.
Yeah. So we covered this on Wednesday. Yeah. So I won't repeat Wednesday's lecture. Yeah. So that we drew chromosomes. We talked about different yeast types. We talked about Neurospora, Saccharomyces cerevisiae, how they can have an unordered pair, an ordered pair. Yeah, we spent a lot of time going through this. Okay, and we talked about how they can undergo sexual reproduction versus asexual. Okay. So does everyone see how you can look at this, these columns of alleles, and you can see the alternating pattern versus the stack pattern, where you have all your homozygous dominant together, all your recessive together, sorry, dominant alleles, recessive alleles together. So those first two classes, we can definitely say no crossover between the gene, right, and the centromere. Whereas these other four classes, we know there's been a crossover event in that little window that we're looking at.
Even though the last two are swapped, exactly 8, 3 days falling.
Yeah, so that's where you get these weird patterns, because they still keep, so really what this is because you've duplicated everything after mitosis. So now it's like you've swapped them, you've flipped one of these over essentially, but they still pack them into little by twos, going by twos by twos. The way they end up ordered in this 4, there's still a version of a crossover event between, yeah. And the way you guys will see that too is to double check is by looking at the numbers. So you should have 4 classes that are all fairly small numbers and somewhat equal, right? 9, 11, 10, 12. And then you should have 2 classes that are much higher in number that represent the ones that did not have a crossover between there. Okay, so what we do is we're going to focus on kind of similar, but also to our 3-point recombinant mapping, where we focused on the recombinants and added them up pair by pair. Remember, we went 2 genes by 2 genes by 2 genes and add up all the places where we see recombination, so we can get map units on distances between genes on the map. So you're going to do a similar technique here, where you're going to focus on those that have that crossover between the gene and the centromere. And kind of, for right now, we're going to ignore those big parental, in a sense, or no crossover between the gene and the centromere classes. So first thing you would do is look at your chart and identify which have had a crossover between the gene and the centromere, and then which have had a crossover between the gene and the centromere, and which have not. Kind of block those out. And then you're going to add up, because ultimately we're trying to tell centromere to gene A distance, right? We want to know what this distance is. That's what we're asking. So you're still using that concept of frequency of recombinations being kind of equal to map units. We're converting a frequency to a map unit. That's essentially still in that 1-to-1 ratio. Okay? So all of those that have had a crossover, you add those up. So those have had a crossover between the gene and the centromere. So it's kind of the same thing as us looking at 2 genes, distance between 2 genes, except for the centromere, as I said on Wednesday, is not heterozygous, so we can't really track it in terms of tracking meiotic products by looking at gametes. So the only way this is really done is in this specific organism that we talked about that makes these ordered spore sacs. Okay, it's the only way that you could do this type of analysis. But essentially, you're still asking distance between 2 points on a chromosome. Okay, so we get 42. What do we do with that? How would we get a frequency of recombinants that are occurring between centromere and EA?
Divided by the total.
Well,
Half.
so you're going to take 42 and divide it into, yeah. We said it, but, yeah. So 42 and divided by 300. Okay, and we end up with this 14%. and you multiply by 100 to get it in percent form. Okay, what else do we have? 14.
Or divide.
Yeah, you have to have, you have to multiply by half, divide by 2, whatever makes sense mathematically in your head. You still have to take that 14 and divide it by 2. Why are we dividing by 2? Not 2, we only have one gene though. Why by 2?
Because there's 8.
Yeah, because you've made, when you undergo mitosis, you're doubling your products. So everything we see is a double of the original, okay? Because we started out as a tetrad, now we're in an octad, so you have to divide that there by 2. And because only 50%, the other way, so the way to think about this is if this is a diploid organism, always you have 2 chromosomes, right? So only half of your chromosomes show that recombinant genotype. And so that comes back from our original, if you go back to Wednesday's notes, remember we drew all the chromosomes, we even had different colors, we talked about how crossovers happen. So we focused on the 2 chromosomes, chromatids, right in a tetrad that show recombination, whereas those outside 2 chromosomes, this guy and this guy, are not involved in that crossover event. Just those inside 2 are the ones that have DNA that's exchanging between them. So these end up being recombinants. And the outside are parental or whatever, however you want to, they're the original combination of alleles. However you want to think about that in your head. So whenever you're trying to determine number of recombinants, you have to take that into account. That's why in your book, I know when your homework problems that was due tonight, you end up having to divide by 2 because you always have 2 chromosomes. One which you're assuming is staying normal, staying parental, and the other one which is involved in that crossover event, those inside chromatids that are crossing each other over. And then we're just multiplying that by 2, as you were saying, because it's undergoing mitosis, so we have a full octad instead of a tetrad that's up here. So you take your percent recombinant that you found, and this is the same as and in essence, it's the same as a 3-point cross. If you were working backwards, taking a map and trying to determine how many progeny fell into each category, you would have to take that map distance, multiply it by the total, and then divide by 2 to get each one of those classes, right? Okay, so we end up with 7. So actually, all this stuff just to find out that this is 7 map units between the centromere and DNA.
So the percent disappears as well?
Well, you have a 1-to-1, right? You have, so 1% is equal to 1 map unit.
Oh, okay.
So I could make that additional step if you.
No, that's fine.
Yeah. But you essentially can convert it over to map units, the same way we did with the 3-point cross. Yeah. Where did I get? 2, we're dividing in half, remember, because you have 4 chromosomes, only 2 are recombinant, the other 2 are chromosomes. Yeah.
Would you ever ask for like the recessive allele, or would it be the same process?
Sorry, yeah, so I see what this is gene A, and you can think of it in either format,
Okay.
either allele. It's just the gene itself. We don't care what the allele is in this context, other than figuring out a pattern here.
Okay.
But you're talking about the location of that gene A and the location of, yeah. And the same with the 3-point cross. We're just talking about those gene locations. We're not really concerned about allele type, because that allele big A is going to be at the same, presumably, if you have 2 chromosomes, if you're heterozygous, and you have big A on one of your chromosomes and little a on your other chromosome, we're assuming hopefully the distance between here and here is the same as the distance between here and here. Unless some kind of freakish insertion occurs or something like that, which does happen. But we assume the distance between either allele, it's that gene location, will be the same to the centromere.
And we're only gonna have to calculate for one gene.
Yeah, this is just gene to a centromere.
Well, in the
Yeah.
homework you had, it was 2 genes. You had to calculate.
That wasn't a
this.
tetrad. Okay, that was for the unordered. Yeah. Yeah, so unordered, the little sack one that we drew that was in your homework, don't worry about that for the exam. Yeah, I was going to cover it, but I'll say more in the video. And that doesn't mean we're gonna like start in, we'll just start in on new, we'll just leave it behind and wouldn't worry about it. Doesn't mean we're gonna necessarily revisit it after the exam either. So does this ordered way of determining centromere to one gene distance make sense? Yeah.
So, actually, no, I don't know.
And one thing I would encourage you to do is, as a sort of as a practice, the way we drew out these chromosomes previously and assigned alleles to them, and we talked about crossover, and then we drew, remember we drew what this result would be and everything. We broke apart the chromatids and thought about the alleles on each one. You can do that for all of these types right here. Show how you would recover this order back. Even before mitosis, if you think about the meiotic, the meiosis events that produce this, you guys can do that. You can draw out your chromosomes and be familiar. That'll be a good study tool as well to make sure you understand why these show no crossover between gene and centromere, and this does. We'll start erasing the beginning stuff, and if you think of any other questions about this tetrad analysis, let me know.
So, because of multiple crossovers, this only works up to 50 mappings, right?
And
Because then you're gonna have.
that's usually more of a rule with the multicellular organisms that undergo meiosis more frequently. And you have, that's more of a rule when you're doing a 3-point cross. So 3 location mapping.
So,
Then you really have to be within that 50 map
okay.
units.
Now,
To a certain extent, that still somewhat applies here as well, because the further we get from the centromere, it's going to be much more frequent, likely that you'll have a crossover event occur between that gene and the centromere. If your gene's at the very end of the chromosome and your centromere's here, of course that distance is greater, and you guys all know the greater the distance, more likely we have a recombination event. So it starts to get less accurate.
is there a way that I'm supposed to know intrinsically that those are the ones with no crossovers, or is it based on the numbers?
Based on the numbers and based on this pattern
So,
that we drew out, yeah.
and I guess, if I understand you, this is what you covered on Wednesday, why, okay,
Yeah, we drew chromosomes and drew how you would get
okay.
this pattern, yeah.
So I think the biggest thing that he's confused about is because we drew the unordered.
We drew ordered. We drew chromosomes. I was there. I was awake. Yep, this exactly right here. So you have big A, little a, little a, big A, little a, big A, little a. Yep, that's exactly what we're talking about now.
So then this basically just goes into all of this.
Those stacked spores are a product of the bigger
Okay.
thing, yeah. So I can answer these questions about Wednesday's lecture when we get to the end and we have the additional questions, office hours stuff. All right. So best use of our time right now, and then I can help those folks who are still confused about Wednesday's lecture, is to go through these additional problems that I have, the pedigree and then the chi-square. So for your notes, here's a pedigree.
Mine has been through much, too much. I've seen too many things. I've read too many things. If you know what I mean. I was born in a generation where
And so actually what I'll ask at this time, since essentially this is all review at this point, if you prefer to leave, if you feel like you have other better study things to do, doesn't, go ahead. It doesn't hurt my feelings at all. But I wanted to give those folks who are just done for the evening that opportunity. Your exams on Wednesday, we start at 6 o'clock. I will check the outside schedule. I might try to get here a little early, but technically we start at 6. I'm happy to let you guys hang out and finish that exam until the next class comes in, which will be 8 p.m. So you'll have 2 full hours essentially. Bring your pencil, your pens, your eraser, bring a calculator. The graphing TI-86 tech calculators are fine. That should be all you need. If you need a chi-square table, that will be provided for you. Any other kind of, you'll have room, lots of room. I'll try to format the exam such you have lots of room to work, do some problem solving or whatever, diagrams, Punnett squares on the side, things like that. Yeah.
Will we have some scrap papers provided for me to do some?
I will make a note and provide scratch paper. I wasn't pointing, but yeah, thank you.
Can we bring our own scratch papers if we forget?
I'll I'll probably just get home and be like, no, I'll bring scratch papers. Any other exam kind of questions? So we're pretty much up through that tetrad analysis. This is everything you guys need to know. After that, this is all review, practice, doing some additional practice. Okay, so if you want to please go study on your own, that's totally fine. If not, we're going to try this pedigree problem.
oh, damn. I never pressed the one. ¿Sí? ¿Cuántos quieres? ¿O quieres el blanco? ¿Blanco?
Okay.
So we don't need to worry about lettering the parents?
Hmm?
We don't need to worry about lettering the parents?
No, I'm not gonna, yeah. We'll just refer to them as the parents. Or grandparents, great-grandparents. Yeah, we're not working with them. Okay. assuming that the first generation there is homozygous, and then any of those folks marrying in from the outside are also homozygous. So if I was to throw this pedigree at you guys and ask you, what do you think is the most likely mode of inheritance? What would your thoughts be? Recessive, right off the bat. Why recessive for those
Because
people?
no one else showed it.
Skip a bunch of generations, there aren't that many people really affected at all, so we definitely think recessive. And then is it autosomal or sex linked?
Sex linked.
And then why do you say sex linked?
Because only males are affected.
Only males are affected, yeah. Look at, you guys just got like 3 points already on the exam. 3 out of 80 is a lot. This first exam is only 80 points. So, it's going good. This is going well. We're already doing so well. Now your problem says use the allele H, okay? So H equaling our wild type normal allele. Turns out this is actually hemophilia, a family that has hemophilia. So H is going to be our normal allele. Middle H will be the disease allele. And it's asking you now, let's assign some genotypes. So you can assign genotypes to a lot of individuals, but for practice we'll do 2C and 2D. So first of all, the parental generation, right? So this is whenever, again, I told you guys, look at it, try to figure out what your mode of inheritance is, try to assign any genotypes you might know right off the bat, just to get yourself some kind of focus and a place to start on these types of problems, okay? One of the things I think I see, and we chatted about this in office hours a little bit today, with the genetics problems is sometimes we read the problems or we look at the data that we're given and our mind just goes, and like I'm looking at this, I'm looking at this, I'm looking at that, and you're a very monkey brain, okay? You're all over the place. You're like my 5-year-old niece was at the aquarium this weekend. She was like a crack addict, you know, an old 5-year-old crack addict just going crazy at every single display of animals. So you have to kind of narrow it down. You have to start somewhere, be very organized, be very anal retentive about how you approach these problems. OCD helps here, okay? So have your checklist and follow it. Okay, you really, and so one of the ways to do that, start, even if the question didn't ask you, what do you think the mode of inheritance is? Look at it. Think about who's affected, males and females, whether it's all the generations or what's going on here. And then start assigning genotypes to those that you can easily see right off the bat. Okay. So because we decided on sex-linked recessive, what kind of allele designations would we give this parent here, this gentleman?
Homozygous recessive.
So he's XY, and he has a little H, right? Yeah. And then the female is XX. She does not have the disease. She does not show any kind of recessive phenotype. She shows a dominant phenotype. So we know she has to have one dominant H.
And it says that she's homozygous.
Right. And because this told you this first generation is homozygous, the reason for that is you can't calculate any probabilities about her receiving that allele. How would we go any further unless we had future further generations above her that were represented? So to make it further in the problem, you assume that she's homozygous.